So this video is to give you a little introduction about some of the things we've been working on here at the Welsher lab at Duke University in the Department of Chemistry. And one of the main things we're working on is looking at single molecules, in particular single biomolecules like enzymes, and how they function in living systems. So one of our big goals is to develop new microscopy methods that allow us to lock on to freely diffusing biomacromolecules such as enzymes or even viruses and capture those dynamic 3D processes in living systems. Of course, the reason we want to do that is that these enzymes or viruses, when they interact in living systems, interact with a very complex and crowded and non-equilibrium extracellular environment. So we want to capture the dynamics of those single biomolecules in their full biological context. And at the same time, we also want to measure exactly how different the kind of chemistry and biochemistry is within this complex and non-equilibrium environment. So the first main aim of our research is to develop active feedback, single molecule and single virus tracking, and this kind of allows us to tackle this first idea, which is to lock onto these freely diffusing biomacromolecules, and to evaluate the kind of complexity of this cellular interior. We also have recently developed some vibrational imaging of live cells. Okay, so the first part of our team is Shangguo, Chen, and Stacy, who work on our single molecule tracking. And the idea here is to develop a microscope which is able to lock on to a single molecule and follow it wherever it may go without the need to tether it. So if we have a protein that's diffusing through the cellular interior, we want to have our laser spot, which is exciting the molecule, follow it wherever it may go so we can measure its dynamics in full context. And the kind of overarching question we're going after is, what role does the crowded non-equilibrium cellular interior play in the functional dynamics of these enzymes? And to get at that, we've developed the 3D single molecule active real-time tracking microscope, which we've cleverly named 3D Smart. The 3D Smart uses a rapidly scanning laser focus, which you're seeing in this animation here. And when this laser spot overlaps with a molecule, we get photons. And those photons are read out by a single photon counting detector. And then we actually move the whole sample to lock onto the molecule. And we get trajectories like the one you're seeing here on the right. This is actually a single fluorophore diffusing in solution. So this is the kind of the basis for the experiments that we're doing in the single molecule realm. So we're also expanding these types of technologies to look at viral infection. And this is an effort with the team of Courtney, Xiaochen, Jack, and Yuxin. So the idea here is, now that we can lock on to fast objects moving in 3 dimensions in complex biological systems, can we finally look at the early stages of viral infection? So these are the stages before the virus has even interacted with the cell surface. It has to go through a pretty complex extracellular space. And the question we want to get at is, how do viruses navigate this complex extracellular space to eventually infect cells? So what we can do is we can develop the fluorescent viruses and lock onto them with our 3D Smart microscope, as you're seeing in this trajectory here. So in this little video is a single viral particle. This is a lentivirus locked onto by our 3D Smart microscope. And then we can extend that to track how these viruses interact with cell surfaces, which is what you're seeing in this video here, where this kind of volume indicates the cell surface. So this allows us to actually see how the dynamics of this viral particle change as it processes through the cellular exterior. And finally, Xiaoqi and Sean have focused on the other part of this problem, which is less on the biomacromolecule side and more on the complex cellular interior side. And the idea is to kind of map how the intracellular environment differs from kind of a normal bulk solution. In particular, one of the things we've been looking at is solvation. It has long been known by physical chemists and spectroscopists that you can use nitriles to read out solvation within proteins. I can put a nitrile at a different place in a protein, and it can tell me whether that area of the protein is hydrophobic or hydrophilic, or if there's any hydrogen bonding. And what we've been working on doing that is scaling that up to the entire cell. So we imagine, take this whole cell, scatter nitriles all around, and just start to see how is the solvation different at different parts in the cell. So we want to know is the intracellular water intrinsically different from what we normally think of as bulk or free water that we would just have in kind of in a test tube. So the way we do that is we do hyperspectral imaging of live cells. We do a hyperspectral stimulated Raman scattering microscopy, wherein we can take images, vibrational images of cells at different vibrational frequencies. And this allows us to get the actual chemical properties at different areas in the cell. And if we add in a probe, so we use this near resonant nitrile probe called rhodamine 800, this gives us a large signal, and it also has the nitrile that allows us to read out the solvation. And what Xiaoqi has been able to dig out here is that when we map these nitriles, so this is just an image of the nitrile distribution within the cell, we can see that there is a definite heterogeneity in the degree of solvation depending on the localization within the cell. So we can actually see that the solvation within the cell is much different. And this just indicates exactly how complicated the cellular interior is and how important it is to do our single molecule experiments in this context. So as you can see in the lab, we get into a lot of different things. We have microscopists, we do a lot of coding, we do biology, we even do a little bit of chemistry.